Hello, and welcome to another episode of the Evolutionary Hologenomics Podcast. Our guest today is Associate Professor Anton Alberti. Anton, thank you so much for joining us on this episode. Would you say some words about yourself?
Well, thanks a lot, Anna. Yes, I am Anton. I am Associate Professor here at the Globe Institute, and I am leading a research group of 16 people. We are researching on how animal microbiota interactions affect multiple ecological, evolutionary, and applied processes and general aspects of animals.
That sounds like a lot of people and a lot of interesting things to do. So today we picked one of those projects that has just started called 3D'omics. Would you tell us what it is in a nutshell?
Well, 3D'omics is a European H2020 project that will be running for the next 4 years, aiming at developing a bunch of new technological advancements to reconstruct animal microbiota interactions at the microscale and in 3 dimensions. So it sounds complex, but we will try to push the limits of our genomics technologies that we have for characterizing these animal microbiota interactions, and by combining different sources of knowledge, different techniques and expertises from different fields of biological sciences. We will have structural genomics, we will have metagenomics, we will have animal physiology, microscopy, and many other approaches that we will try unify to create a new framework, methodological framework that will be then implemented to address different questions that are relevant for animal production industry.
It does sound very, very ambitious and complex, but we will try and break it down a little bit during this episode. So you mentioned H2020 or Horizon 2020. Those are very big projects. Could you tell us a little bit for either those people who have not applied but would like to apply for something like this, or for people who are actually not scientists, just to have an idea of the time scale and also how much work and how many people it involves just to prepare an application for such a huge project?
Mm-hmm, sure. So the European Union has this broad and large program for funding research projects that encompass usually more than 10 or even 20 partners from different countries. There are work programs that are released with specific topics to which different consortia can apply for getting decent amounts of money for doing research. In our case, it is research and innovation action. There are different types of projects, and some are more applied, some have more basic aspect for developing research technologies. And in our case, that's the type of project we have. We have some technological developments and then implementation in applied areas. This project is included within the program of food security. So the European Union is putting quite a lot of money to investigate, to research on how we can improve production of foods, the security of our production and consumption. And in our case, our project will last 4 years. We have a budget of 10 million euros, and we are a consortium that encompasses, I think, 10 different countries. And this is this was created through a rather complex process where we first started sounding out this idea with some close partners, and then trying to identify which were the key knowledge gaps or gaps of expertises that we had, and then try to contact relevant people. And that's how we created our consortium. I think I have an overview of yeah. So we are 13 partners in our case from 11 different countries, and that involves 6 universities, 2 research centers, and 5 companies. So the universities and the research centers will be mainly involved in developing the technology and in creating this new framework to later apply this technology on aspects that are of relevance for the industry and for the sustainability and of food production in Europe and globally in the end. So the process of applying for this project is quite long. In this case, it was a two-stage application. So we had to apply for a shorter version of the project initially. Then after 6 months, more or less, we got a positive decision, and we had to apply for the actual project with a rather large application where we need to detail all the information on what kind of research we will do, how this research will be implemented, and then how this information will also be disseminated across the relevant stakeholders, scientific and industrial stakeholders, how the results will be exploited in the future, and how all this information will be also reach the public audience at different levels. It could be also industry stakeholders, but also the general public, or even kids, the schools, etc., to show what kind of research we are doing in Europe.
Right, so there are many, many moving parts and many pieces to this puzzle. So not only the different partners in different countries doing with different expertise, doing the various parts of the work, but then there's also the very important part of communicating your results to the scientific community, as well as beyond to industry and the general public. So that's a lot of work to do. So does everyone work at the same time, or did you divide the work across these 4 years? And people are in very different countries. So how will they all collaborate and work together?
Well, of course, the tasks within the project are allocated to different partners and people. I think you have an overview. Yeah, so this is how we structured the project. We have 4 main sections, and one of them is dedicated to management, and then dissemination, exploitation, and communication. And we can see that we have a work package that is dedicated to this communication topics. So this will be mainly centralized here at the University of Copenhagen, but all the partners will be also involved in different activities. There will be outreach activities, but there will be also training activities, and there will be also other kind of networking events that we will be organizing to make sure that the new technology that we will be developing will reach the relevant stakeholders, and we will also hear from them for further developing our technologies, and then to find also new possibilities to implement it in a way that can be useful for getting knowledge that we don't really know nowadays. Then we have 3 main sections. The project is divided in 3 main sections. There is one part, as I was mentioning, about technology developments, and then we will be implementing that technology in 2 different sections. So the 2 different work packages. One will be focusing on health of chickens and turkeys. So we will be running 3 experiments and then applying our technology in the animals that we will be sampling in these experiments. And there will be other set of experiments applied to swine, and where we will be mainly dealing with nutrition-related issues. And once we generate all this information and we process this data, there will be a last section where we will assess the technology, how are the limits of this technology, what are the potential for further developing it in the future. And we will be also assessing what could be the impact this technology will have in the future in research, in the industry, but also in the environment. Because if we manage to understand animal microbiome interactions in a higher level of detail, we might be able, or we should be able to develop new strategies that will be more sustainable and will contribute to really improve how agricultural production in general is done nowadays. Because the technology will be very, let's say, very agnostic in the case of the taxa that we will be working with. The technology would be applicable to, or should be applicable in the future to any animal model, but also to plant models to study what are the interactions that happen between plant roots and the microbiota that are in the nearby soil, etc., etc. So we really hope that this technology will make a difference to improve research and production processes in the future.
Yeah, so there is a method to the madness, and all of this is nicely organized in 9 work packages, which are basically groups of tasks to achieve different things. So that's good. That's the structure. And can you tell us now a little bit more about the actual work and the research and what you guys intend to discover? So you mentioned that there were some gaps in knowledge. So what gaps are those, and what will you do to fill them in?
Well, nowadays, the way we usually analyze microbial communities that are associated with complex organisms, this could be animals or this could be plants, it is done in a way in which the spatial structure is completely missed, because we get samples and we homogenize these samples. So it is not the optimal way to understand how microbial communities or microbes within these communities are interacting with each other and are also interacting with the tissues of the plants or the animals. So what we are aiming to do is to develop a technology to overcome this limitation and to generate genomic data and metagenomic data that preserves this spatial information. So here in this picture, yes,
just
Anna.
Stop you there. And so you've met, we with this podcast aim to sort of clarify some bits of jargon. So when you say that usually things are homogenized, and now you intend to separate things and see where they are. So what does homogenized mean?
Well, when we get a sample of intestinal content or a fecal sample or a root sample, what we usually do is to mix very well all the material so that then we extract the DNA or the RNA or the metabolites that are there using different chemical and physical procedures. But all the DNA, RNA, metabolite extraction procedures that are nowadays applied require of this thorough mixing of the sample. So the microbes that were there when we sampled the material will be there, but their spatial information will be completely lost because they won't be linked to each other anymore. They will be all in a homogeneous mass that when we reconstruct these communities. And this is a pity because there is a lot of useful information in the spatial arrangement of the different biological elements. And this is what we want to overcome. And we have ideas. We came up with some strategies where we could use technologies developed in the field of structural genomics, where they try to reconstruct how the DNA molecules inside the eukaryotic cell nucleus are folded and what kind of three-dimensional structures they have and how this affects the expression of the different genes, etc., etc. We want to extrapolate those technologies to the microbial communities and microbial communities that are next to animal epithelial cells, so that we can try generate genetic information of different bacterial cells, different eukaryotic or animal cells by preserving their spatial information. And that way we would know whether two bacteria that are closer to each other are interacting in a certain way, if they are expressing some genes that other bacteria that might be genetically very similar but are not in the same conditions, so they are not close to, they are not within the mucosal layer that might have a completely different biochemical properties compared to the lumen of the intestine, so the central part of the intestine. Then we would like to cover all this information and this way reconstruct with a much higher level of detail. In this case, we will be talking about a spatial scale of a few micrometers. Usually when we sample these animals and we get samples that are in the scale of centimeters or a few millimeters. In this case, we will go 1,000 times lower scale and try to see how these spatial patterns change and how this could be potentially linked to the performance, to the health, and to other biological traits of the animals.
Right. So this is basically the name of the project in a nutshell, 3D. So you want to see where all the bacteria and all the cells are and what are they doing. And then omics is just the different layers of omics. So you will be looking at DNA, so that's the genes, RNA, so that's the expression of the genes. And you also mentioned metabolites. So can you elaborate just a tiny bit on either of these, just to make sure our listeners are following with us?
Yeah, so we will reconstruct virtually the entire genetic information that eukaryotic or animal cells and prokaryotic and bacterial cells contain. This genetic information contains genes, so we will also be able to annotate, to predict genes and annotate genes, and then quantify also what is the expression of these genes by analyzing the RNA. Then these RNA sequences encode for proteins that participate in different metabolic processes, and these proteins in many cases act as enzymes that catalyze processes that's to get some metabolite and convert into another metabolite that will have a different function. So through the metabolomic layer, we will be able to capture these metabolites. Through the transcriptomic layer, we'll be able to quantify the expression of the different genes, and through the genomic layer, we'll be able to characterize what is the entire landscape of the basal information, genetic information that is present in the system. And then we will integrate all these three layers of information that will also have different spatial resolution. So there will be some challenges there to understand how the interactions between microbes and microbes and host cells happen, and how these interactions have impact the health, the performance of the animals. In the different trials that we will be running, there will be different challenges. So we will be challenging animals with a viral pathogen, a bacterial pathogen, as well as a protozoan pathogen, different experiments. All these pathogens are known to be detrimental for production processes, or could be problematic for the health of the produced, or the safety of the produced food. So we will see how different management practices through probiotics or through vaccines affect the intestinal landscape in which the microbes, the pathogens, and the animal tissues interact. And this way, by better understanding how these interactions happen, we hope that we will be able to develop new and more optimal processes or procedures, strategies to improve and to overcome these health and performance issues that we nowadays have.
Yeah, and that is very, very multidisciplinary. So for people who think, you're a scientist, this is all biology, actually this involves so many different fields of knowledge. So this is why also, I guess, you have all these different partners. So not only from academia and industry, but also so many different bits of knowledge. So I assume if you want to see something, and if you want to stain these bacteria and see what they are and where they are, then you need very powerful microscopes and imaging techniques. So that is one field of knowledge. Then people who will be extracting and analyzing DNA and RNA and metabolites, that requires different knowledge and skills. And then all of this, because it's so large scale, requires powerful computers and a lot of bioinformatics analysis. So is this more or less the different expertise and knowledge that you assembled for this project?
Yeah, that's absolutely correct. So it is an extremely multidisciplinary project. So I would say that it is an interdisciplinary project, because we will be integrating all these different expertises. And I would say that's the main feature of this project. It is a courageous project, a brave project, in the sense that we will be exploring a domain that nobody else has explored so far, at least at this level of detail. And that requires a lot of different expertises, those that you have mentioned, but also very important, the animal sciences, animal scientists that really are interested in developing new solutions to improve all these processes. And the technology that we would be producing would be a tool that in the future would be able to provide more detailed information so that animal scientists could improve this production process. And of course, this includes animal scientists that are in the academia, but also animal scientists that are in companies that are producing different feed additives, feeds, could be also health treatments to improve the safety and productivity and sustainability of the agricultural production in the end.
Yeah, that is very, very ambitious. And so nothing like this has been done before.
Well, there are some studies that have approached that have started to explore this domain of generating, let's say, microscale molecular data to get a more refined view of how microorganisms are organized across the intestinal landscape. I have a couple of examples here. We have this rather recent paper by Shed and colleagues that was published in Nature Biotechnology, where they used an amplicon sequencing-based approach after embedding small intestinal sections within beads, and that was one initial approach for getting this spatial information. There is another paper by Riva and colleagues in Nature Communications that did something similar based on laser microdissection, and they also saw that there is a spatial structure of how the microbial communities are distributed across the intestinal landscape. But we will, in our project, we will try to build on all this knowledge that these colleagues generated a few years ago and extrapolate this into multiple omic layers on one hand, and also to include the animal tissue part as well. So it won't be limited to the microbial communities, but will be also including information of the tissues on which these microbial communities grow in. So it's the substrate in the end for these microbial communities, because most of the microbes that are in the intestine will be close to the mucosal barrier, that is the envelope, the end of the intestine. And by knowing how these different microbes occupy different micro niches with different properties, that could be different pH, different oxygen, etc., we will be able to understand much better why some bacteria appear in some conditions, some others don't. What are the different functions that different bacteria really do under different conditions, because we might have the same bacteria here and there, but we might be doing completely different things. If the pH is pH 7 or pH 4, if the oxygen is higher or lower, or if they are close to other bacteria that are producing some metabolites that they use as substrates, and then they convert into something else. So we will try to get to this level of resolution to see whether we can go beyond a mere characterization of what is there, and really try to understand what each of these players are doing, depending on where they are spatially located.
So that involves a lot of work in the lab. So from the actual trials to then collect these samples, prepare and process these samples, and then finally analyze them. So you mentioned that the different partners are with different expertise are across Europe and a bit beyond. So the idea is that these samples are going to be sent between, or the data, because it seems like a lot of coordination to do and to make the most out of one sample, right? Because from one sample, then you want so many different kinds of information. So it's like you have the sample, you break it in bits, but you label it to make sure that you know where the pieces of the puzzle go. And then at some point, you're going to put them all together again. So how does that work?
Yeah, exactly. It is an extremely integrative project in that sense as well, because there will be some partners that will be running the trials, the animal trials. There will be a very intense sampling of these animals where the trials will be conducted. Then these samples will go to another place where the processing of the samples will happen in the lab, or will start in the lab. There will be part of embedding these samples, cryo-slicing these samples, micro-dissecting the samples, and then different sample types will be distributed across different partners to generate different omic layers. Some will be working on metabolomics, some will be working on metatranscriptomics, and then all this information, along with other imaging data that we will be also generating, will be integrated by another partner that will be in charge of reconstructing these three-dimensional structures. And once we have that, then will be the time that microbial ecologists will try to start really finding patterns that are ecologically meaningful and can give us really insights into what is happening. And once we have that information, then will be the time for animal scientists to see how these animal microbial interactions might be really affecting the performance of the animals. So it is a long journey, and we know that time-wise we will be really constrained, and that's why now in the beginning of the project we are really working in making sure that all this communication and management of samples, of data is really very well defined, as well as the tasks that each of us will be doing. So we will have everything, all the different tasks that needs to be done will be very well defined. And because everyone needs to know when each of the different tasks will start, will end, who will be transferring what kind of information, data, samples to whom. So this is one of the main challenges. And this is sometimes, I would say, probably the dirty work that usually will be hidden because it won't be shown anywhere, because it's not really something that nobody really likes or enjoys listening, but it is probably what takes most of the time of the research project. And it is extremely important to make sure that all the databases are properly structured, all the access rights for different kind of information is also very well defined. And then the information and data flows are very well defined, because we are not talking about small amounts of data. We are talking about terabytes and terabytes of information that need to be transferred through the cloud. And this also takes time. We need to be prepared also if there is any problem with freezers and some samples are lost. We have backup samples. The same happens with the data. If there are some data now, for whatever reason, a server is completely wiped out, then we need to make sure that these data are backed up somewhere else, etc., etc. So we started with the project in September. We are already in November, and we continue the polishing of these procedures to make sure that when the actual processing of the bulk of samples happens, all these processes are properly defined. And that way, I think that it's going to be much easier to really succeed and achieve the goals we have set in the beginning of the project.
So that's a very, very busy start to make sure everything is planned and all the different components are there. And then you need a backup of the backup of the backup just to be absolutely sure. And you'll be generating a massive amount of data. So yeah, good luck in 4 years.
Thank you.
Let's see how it goes. And so do you foresee, because 4 years seems like a long time, but hearing you talk about all the different things that need to be done, suddenly it doesn't seem like it's such a long time. So there will be a lot of things to be achieved, and hopefully then it can be used. It doesn't matter what species or what sector. Do you foresee that then people can actually not only use your results to improve techniques or animal production and so on, but also the actual data that comes out of this project, or you are intending to analyze everything and kind of just finish there?
No, so of course, we won't be able to make the most of all the data we will generate in 4 years. We are completely aware of that. So our aim will be first generate this, develop this technology, then assess what percentage of what fraction of this technology can be really implemented at a larger scale, at a higher throughput for the second part of the project. Then there will be also a parallel development of the technology that will happen, but we also need to balance between the resolution that we get and the amount of samples we can generate, balancing the time, the cost, etc., etc. So once we have assessed everything, then we will generate some data that we'll be able to analyze. There will be a fraction of data that we won't be able to analyze, and many other people will be able to have access, to get access, and continue developing new techniques, developing new algorithms to better analyze this information. So we are planning to publish all this information and as an open access so that other researchers from anywhere else in the world get access to this data and help us really push all this, let's say, new fields of science ahead, because this is just the beginning. And I'm pretty sure that a lot of really exciting things will happen. Yeah, but of course, as you mentioned, so this requires the joint effort of a lot of people, and that requires also a lot of people working in parallel. So some people will be developing laboratory methods, some other will be developing bioinformatic methods, other will be developing statistical methods, and we will be also recruiting people for that. So we have actually prepared a project presentation event that will happen in November the 30th. So in a couple of weeks, slightly over a couple of weeks from now. And this is an event that we will organize through Zoom. So it will be open and mainly focused on researchers, mainly young researchers that are really eager to develop new technologies. We will be presenting what we are planning to do from the bioinformatics laboratory perspective, imaging perspective, etc., etc. And we would like all these researchers that might be interested in these topics to hear from us, to talk to the different partners that will be involved in the project, because in the future there will be many opportunities, job opportunities within 3D'omics, and we would like the most relevant people and the people that are really most skilled and motivated for developing this kind of research to know firsthand what we are planning to do. And then hopefully this will end up in expanding, let's say, the 3D'omics family to people that right now might be somewhere else doing something else, but might be working with us in 1 year or 2 years from now.
There you go. You heard it here first. So if you are looking for to join this adventure that is very ambitious and multidisciplinary, for sure there will be opportunities to learn a lot from many different people. Or if you have a friend or a colleague, then consider joining this event for sure. Where can people join? Is there a website? Do you know the address? Where can people sign up for this event?
Yeah, so we have a project website, 3D'omics.eu, and we are also present in social media. So if you search for 3D'omics, then I'm pretty sure you will find some information, and our project manager and communication manager will be putting all this information that's available to the public. So yeah, mainly 3D'omics.eu, most of the information will be there, and we will be adding more and more information of the project, the different partners that will be involved in different practices, and we'll be also adding some news in the future. So yeah, I think that will be the main place where to find information about this project.
All right, so make sure you don't miss it and join this opportunity to hear more about the project, and also ask the various people involved in the project some questions. So that's November the 30th from 2:30 to 4 p.m. CET. So I guess that's a good choice of time as well. Hopefully you can reach a few different continents, some people waking up, some people finishing their day of work.
Exactly, that's the aim. Yeah, we would like to have the broadest coverage possible. So that's why we decided to organize this event at that time. So that's the Danish time. So it would be, if people have problems with the CET, just look for what's the Danish time.
Danish time, yes, that works too. So thank you very much for introducing us to this very interesting project. We'll be following along to see what discoveries you guys do and what new data you come up with. Would you like to say any final words about the project?
Well, I would like to thank you for providing us this possibility to present our project in this podcast. And no, it's just that we are really thrilled and we are really motivated to open new horizons in this research field. And we really welcome researchers that are interested to directly or indirectly participate in 3D'omics, discuss different research possibilities for the future as well, and data sharing possibilities for joint analysis to address different questions. So we are completely open, and I would encourage anyone interested in contacting me. So you will find information on the internet for sure on how to contact me.
So 3D'omics.eu. Go there and find all the information you need and share it. So Anton, thank you very much for joining us for this episode, and best of luck with this complicated and ambitious, but hopefully very fruitful adventure.
Thank you so much, Anna.
After a little break, Evolutionary Hologenomics Podcast is now back on track, and we have quite a lot of exciting new episodes lined up for you. So if you don't want to miss any, make sure you subscribe, and you can find Evolutionary Hologenomics Podcast in all your favorite platforms, whether it's Apple Podcasts, Spotify, and many more. Just search for Evolutionary Hologenomics, and I will see you in the next episode.